Cellular energy acquisition is fundamental to all living organisms. Organisms obtain energy through different mechanisms, which determines where they can live and how they interact with their environment. Phototrophs harness light energy through photosynthesis. They use pigments like chlorophyll to capture photons of specific wavelengths. These organisms must capture light of specific wavelengths to efficiently harvest energy. Too low intensity provides insufficient energy, while too high intensity can damage cellular components. Phototrophs are found in environments with access to light, such as surface waters and terrestrial habitats. Chemotrophs, by contrast, derive energy from chemical compounds through processes like oxidation. They extract energy from various chemical substances rather than from light. These organisms don't require light and can thrive in a variety of environments. Chemotrophs can be found in environments like deep-sea hydrothermal vents, where sunlight never penetrates. This fundamental distinction between phototrophs and chemotrophs determines where organisms can live. It also influences how they interact with their environment and other organisms within their ecosystem. Organisms differ in how they obtain carbon, which is essential for building organic molecules and sustaining life. Let's explore the two major strategies for carbon acquisition: autotrophs and heterotrophs. Autotrophs can fix carbon from inorganic sources such as carbon dioxide. Using energy from light or chemical reactions, they convert this inorganic carbon into organic compounds. In contrast, heterotrophs cannot fix inorganic carbon. They must obtain their carbon by consuming organic compounds produced by other organisms. Examples of autotrophs include plants, algae, and some bacteria like cyanobacteria. Meanwhile, heterotrophs include animals, fungi, and most bacteria. This fundamental difference in carbon acquisition creates the basis for food webs in ecosystems. Autotrophs form the base of the food web as primary producers, creating organic compounds from inorganic carbon dioxide. Primary consumers, mainly herbivores, obtain carbon by eating autotrophs. Secondary consumers, like carnivores, get their carbon by consuming primary consumers. Finally, tertiary consumers are at the top of the food chain, obtaining carbon by feeding on other consumers. This structure illustrates how carbon flows through ecosystems, originating from autotrophs' ability to fix inorganic carbon and moving upward through heterotrophs at various trophic levels. Photosynthesis occurs in specialized organelles called chloroplasts. Inside the chloroplast are stacked membrane sacs called thylakoids, surrounded by a fluid called the stroma. Photosynthesis consists of two main phases. The first phase is called the light reactions, which occur in the thylakoid membranes. The light reactions involve two major protein complexes, photosystem II and photosystem I. Photosystem II captures light energy and uses it to split water molecules. This releases oxygen gas as a byproduct. The splitting of water also releases electrons that flow through an electron transport chain. This creates a proton gradient across the thylakoid membrane. The proton gradient powers ATP synthase, which generates ATP, a key energy molecule. Meanwhile, electrons continue to flow to photosystem I, which uses more light energy to produce NADPH, another crucial molecule for the next phase of photosynthesis. In summary, the light reactions produce three key components: ATP, which provides energy, NADPH, which provides electrons and hydrogen, and oxygen gas as a byproduct. These products are then used in the second phase of photosynthesis, known as the dark reactions, or the Calvin cycle. The dark reactions, also known as the Calvin cycle, occur in the stroma of the chloroplast. Unlike the light reactions, they don't directly require light. The Calvin cycle consists of three main stages: carbon fixation, reduction, and regeneration. In the carbon fixation stage, carbon dioxide from the atmosphere is incorporated into organic molecules using the enzyme Rubisco. The ATP and NADPH produced in the light reactions provide the energy and reducing power needed for the reduction phase. Through these reactions, carbon dioxide is converted into glucose and other carbohydrates. The light and dark reactions work together as a complete photosynthetic process. Light energy drives the light reactions to produce ATP, NADPH, and oxygen. The ATP and ADPH then power the dark reactions, which consume carbon dioxide and produce glucose. The dark reactions return ADP and NADP plus back to the light reactions, completing the cycle. Through this two-stage process, plants convert light energy into chemical energy stored in the bonds of carbohydrates like glucose, which can be used as fuel for cellular processes. Cellular respiration is the process that extracts energy from glucose to produce ATP, the energy currency of cells. The process involves multiple stages, beginning with a glucose molecule and ending with energy in the form of ATP. Cellular respiration occurs in four main stages: glycolysis, pyruvate processing, the Krebs cycle, and the electron transport chain. Stage 1 is glycolysis, which occurs in the cell's cytoplasm. This process breaks down one glucose molecule into two pyruvate molecules. During glycolysis, the cell initially invests 2 ATP molecules but gains 4 ATP, resulting in a net gain of 2 ATP. The process also produces 2 NADH molecules and can occur with or without oxygen. In stage 2, the pyruvate molecules enter the mitochondrion. Each pyruvate is converted to acetyl-CoA through a process called pyruvate decarboxylation. During this conversion, each pyruvate loses a carbon atom, which is released as carbon dioxide. Each reaction also produces one NADH molecule. The third stage is the Krebs cycle, also known as the citric acid cycle, which occurs in the mitochondrial matrix. In this cycle, each acetyl-CoA molecule is completely broken down. The cycle produces 3 NADH, 1 FADH2, and 1 ATP for each acetyl-CoA molecule. The cycle also releases 2 carbon dioxide molecules for each acetyl-CoA. Since each glucose molecule produces 2 acetyl-CoA molecules, the Krebs cycle runs twice for each original glucose molecule. The final stage is the electron transport chain, which occurs on the inner mitochondrial membrane, or cristae. NADH and FADH2 molecules from previous stages donate high-energy electrons to the electron transport chain. These electrons flow through a series of protein complexes embedded in the membrane. As electrons move through the complexes, protons, hydrogen ions, are pumped from the matrix to the inner membrane space, creating a proton gradient. At the end of the chain, electrons combine with oxygen, the final electron acceptor, and hydrogen ions to form water. The proton gradient powers ATP synthase, which acts like a molecular motor. As protons flow back through ATP synthase, it uses this energy to produce ATP. Overall, cellular respiration produces about 30 to 32 ATP molecules from a single glucose molecule, making it a highly efficient process for energy extraction.